Okay. Okay. Now, I missed out on that. Okay. So again, G1, most cells spend their time in it. S phase, they duplicate their DNA. G2, they get ready with all the energy and resources they need for mitosis. And then mitosis is prophase, where the DNA condenses. The nuclear envelope disappears. The chromosomes line up along the middle of the cell. These are homologous chromosomes. One set goes to one side, one set goes to the other during anaphase. And then telophase, the nuclear envelope reforms, and the cell actually has a new cell membrane, and so you end up with two daughter cells. Now, in cancer, of course, we've got a situation where, for whatever reason, a cell continues to divide. Normally, it's stopped with tumor suppressor genes that recognize when a cell is kind of getting out of hand. There are genes that, let's say, when they get stuck on, instead of, in terms of regulation, being turned on, expressing, and then not expressing the proteins anymore, they continue to divide. Those are called proto-oncogenes, which I don't really care for in terms of the word, because oncogene means tumor-causing gene, and these are genes that they're normally not causing tumors. They're just part of the regulatory framework of telling cells when to divide. But you can get a situation where they kind of go a little out of control, and then they become oncogenes. In other words, they get jammed on, let's say, or off, and cells start dividing wildly. Now, even then, with the tumor suppressor genes, like BRCA1 and 2, those cells can be stopped during the different checkpoints. There are also genes that regulate cell replication, and those are tumor suppressor genes. And unfortunately, if you have a mutation in one of those, that means your tumor suppressor system is not really working well. And so normal cells that start replicating wildly normally would be stopped by that system. They're not. And so they continue on. So if you have a mutation in BRCA1 or 2, higher risk of breast cancer, number of ovarian cancer, actually prostate cancer, it turns out, for some. So, and then of course, in terms of types of cancers, if you just have a bunch of cells dividing but they don't invade surrounding tissue, they're just benign. But if they do invade, normally what happens is there's this thing called contact inhibition. So when a cell divides and it comes up against another cell, it stops right there. That's contact inhibition. When there's a loss of that, they keep dividing and invade a cell, the surrounding tissue. That's a malignancy. That's a cancer. It doesn't mean that it could be a fast-growing cancer. It could be a slow-growing cancer. Benign tumors can be really bad, like pineal tumor in the brain. But for the most part, malignant cells, malignant cancer cells are the worst. And then also, normally what happens is the cell membranes have proteins that make them sticky. So these cells stick to each other. In other words, they don't go floating off and traveling around. Well, if there's loss of anchorage dependence, then you end up with metastatic cancer. And the cells start traveling either through the blood system or the lymph system, and then you can have multiple tumors all over the body. Okay, most cancer is not the result of just one mutation. It's called the multi-hit model, and you might start out with one mutation, and then another cancer cell while it's dividing gets another mutation, and they start to build up, and finally you get to the point where, let's say, everything falls apart and they start replicating wildly. So that's called the multi-hit model. You can't catch cancer for the most part from other individuals. You can't as humans. If you're a Tasmanian devil, you can. But you can certainly inherit genes that make it likely that you will develop cancer, like retinoblastoma. It's an eye tumor. If there's an inherited version, it's like 90% chance that you're actually an individual will develop eye tumors. And it usually starts when they're very young and they lose both eyes. You should know, different kinds of cancer treatment and detection. Things are happening very rapidly in the treatment sector, especially with immunotherapy. But there's a lot of cancers that just don't respond. At this point, like pancreatic cancer, nothing really stops it. Okay. Now remember, carcinogens are anything that causes mutations that cause cancer. So whether it's UV radiation, too much time spent in the sun, and you end up with a melanoma of the skin, or some toxin that you grew up around in a site where they didn't clean up things properly. Certain plants are mutagens or carcinogens, so if you eat those, they cause mutations in cancer. Okay. And then there's a bunch of questions, of course, that you should know how to answer. Tumors that do not invade tissue are called what? Are they malignant or benign? Benign, right. While tumors that can invade nearby tissue are called malignant. Okay. Cancer cells that have lost the property of what can leave the original tumor site and spread to the body in the process called, okay. So what is it that you've lost that allowed tumor cells to travel? Anchorage. Right, anchorage independence. Or anchorage dependence, you lose that, and then that's metastatic cancer. Actually, classical chemotherapy pretty much kills everything. It's just that it might kill some of the cancer cells faster than the rest of your cells. And that's why people usually get very, very sick. Now, the tumor suppressor system works because it recognizes damage to cells. And then it causes those cells to commit suicide. So, in terms of radiation therapy, in what case does radiation therapy not actually work? In other words, what kind of cancer cell mutations? Well, they don't work when it concerns the tumor suppressor system. So as long as your tumor suppressor system is working, then it'll detect damage to the cells from radiation. It'll make them kill, self-destruct. But if there's actually a mutation like BRCA1, then radiation usually doesn't work for those because you have to rely on a system that actually causes the cells to self-destruct. Okay. So let's see. So you understand what a biopsy is and why it's often important to get multiple biopsies from the same area, because there's a lot of variation. Usually a lot of cancerous tumors have many mutations that are different, and so you have to actually be able to tell. You might get a sample where it shows very few cancer cells, or and then another one nearby that does. Smoking is a risk factor only for lung cancer. True or false? False, right. It obviously, mouth cancer, throat cancer, esophageal cancer, also colon cancer. So tobacco is not good for you. HPV is human papillomavirus. You can get cancer from viruses. They affect the cellular machinery, and so if you get a sequence that's inserted into one of your cells, it might cause it to become cancerous. Oh. Okay. Are gametes haploid or diploid? Gametes, like sperm and egg? Haploid, exactly. So these are germline cells germline cells are diploid, but they're making haploid copies of themselves during meiosis. Okay? So gametes are always haploid. Whole organisms are diploid. So when you have a sperm and an egg, a sperm fertilizing an egg, that restores the diploid state of an individual. You should know kind of the sequence that meiosis undergoes in terms of there's meiosis 1 and meiosis 2. It does start out very much like mitosis, except there's some extra steps that are involved, like crossing over, so you have more genetic variation that can be passed on to offspring. Homologous chromosomes, they're always diploid, remember? You've got 2 chromosomes, one set mostly from mom, one mostly from dad. We'll get into this more, but why have sex? It's a matter of diversity. If you have an organism that lives in a very stable environment, sexual reproduction is actually not adaptive. You can produce twice as many offspring that are clones of themselves. But if you live in an environment that changes, either time and space, you're likely to have certain genes that work in one environment better, or certain alleles than another. Okay? So that's kind of why there is sexual reproduction. It actually allows organisms to more likely to have alleles that in some cases will help them survive and reproduce in different environments. Okay. You should understand sex chromosomes. You should understand autosomes and somatic cells. Those are the ones that are not part of the germline. They undergo mitosis, but they don't undergo meiosis. You should understand the difference between homozygous and heterozygous. So let's say you have the alleles for hemoglobin. There's a normal type, and then there's the S, the sickle cell type. An individual that's homozygous for the normal type has functioning red blood cells that carry a full load of oxygen. But they're susceptible to malaria, so in an area where there's malaria, they're susceptible to infection and there's lower fitness. Heterozygous individuals, on the other hand, have sickle cell trait, not disease, and they're resistant. And then homozygous recessive, they're lethal. Heterozygote advantage, occasionally you get that, and that's like sickle cell trait, where you have one copy of each as a heterozygote, and that has the highest fitness when there's malaria around. I guess that's good enough. There will be problems with you figuring out whether something it produces, how many homozygous recessive babies it produces, in terms of whether a mom, let's say, is a carrier or something, and the dad isn't a carrier or whatever. So go over the Punnett square problems, and there'll be enough on the test areas for you to actually do those.
The Punnett square problems are on top hat or on?
There's some on top hat, and there's also some on here.
Okay.
Okay?
And the answers are there for them too?
What? Yeah.
Okay.
Yeah.
I had a question about top hat. If you already got the 50 points for it, do you have to continue doing them, other than for studying purposes?
No. If you've got all the 50 points, yeah, you're set.
Okay, thanks.
But keep doing them.
Yeah, for studying, yeah.
Okay.
I had a question. Could you explain contact inhibition?
Okay. So, if a cell maintains contact inhibition, that means if it reproduces, it stops when it comes into contact with other cells. So that's contact. Right. But if it loses contact inhibition, it continues to replicate and invade surrounding tissue.
Okay. Are you going to send this that you went over as an email, like the study?
Oh, yeah, I am. Absolutely.